Okay. Welcome, everybody. It is my pleasure on behalf of the HL7 CodeX Fire Accelerator to welcome you all here today. And to folks who are watching this recording in the future, hello from the past. I hope the future is treating you well. We have a feature presentation today from our friends in the GenomeX Pathogen Genomics team, and I'll be introducing Emily and Akash in a moment. And we also have a short presentation from our colleagues in the Pharmacovigilance and Risk Management team here. And Kelly and her colleague Patrick will be making a short intro and update to to their use case. Before I jump into that, though, I'd like to just do a regular introduction to CodeX for anybody who is perhaps attending their first community of practice, just to understand a little bit about the context of what we're doing here today. So first off, I'd just like to share that an HL7 Fire Accelerator is a project that's assembled by HL7, the standards organization, with a goal of applying digital health standards to very real healthcare domain challenges. And so each of our accelerator colleagues has their own healthcare vertical to attend to, whether it's public health or research or payer data exchange or whatever it might be. Ours happens to be clinical medicine, and we started with oncology and have since expanded into cardiovascular health and to genomics as well. We're going to be focused on genomics in today's presentation. As I mentioned, we started in oncology. So we have three domains. The first is oncology, the second is cardiovascular health, and the third is genomics. Each of these teams is comprised of a number of projects. We call these projects use cases. And this was a screenshot I just took this morning from our Public Confluence website. I'll be sure to put the Public Confluence website into the chat for anybody who wants to look it up. And you can learn more about what each of these projects mean, the kinds of problems that they're attempting to solve together, and in many of the cases, work products that have emerged from this important collaboration. And finally, CodeX is, of course, our purpose. CodeX is the work that we do, but CodeX is very importantly our members. And so this is a fairly recent screenshot. This is a constantly evolving graph that shows the breadth and depth of organizations that invest in the work of CodeX. And we're very grateful for each of these organizations at each of their membership tiers. And if you have any questions about getting more involved in CodeX, please do reach out to me. I'm always happy to sit down with folks and share more about the value of CodeX and how it can help you in your organization, and also to let you know how you can become more engaged in CodeX. And that's all I need to share, I think, about our context, what brings us together. We host these community of practice calls approximately every 2 months in order to bring together a broad audience of members and folks from the public who want to know more about CodeX. These sessions are recorded. We upload them to YouTube. We hyperlink them in our Confluence site. And we do tend to get a fair bit of follow-on viewership of these recordings. And so I look forward to engaging with you as a potential member and answering any questions as we move forward. So I'm going to stop sharing now. And I'm going to invite my colleague Emily. Oh, sorry. I'm going to ask Kelly first. Kelly, would you please introduce yourself and the use case that you and your colleagues are leading, and just give us a brief overview of it. We want to make sure that we save most of our time today for Emily and Akash's presentation.
Sure thing. So first of all, thanks, Emily and Akash. Pat, welcome as well. I'm Kelly Petzel. I'm the use case coordinator for the Pharmacovigilance and Risk Management use case, which is a continuation to the REMS use case that we did close. So as a quick refresher, REMS is a safety program that the FDA requires for certain medications. The intent basically is to ensure that the benefits outweigh the risks of that medication, but the operational reality is often very complex. So when we first started, we were just trying to fix one very specific problem, the complexity and fragmentation of REMS, right? But as we built the technical foundation, foundation, something bigger happened. And we realized that REMS wasn't just the ceiling, it was a doorway, right? Because the movement we're creating proves that you can streamline one of the most complex safety workflows in healthcare, and you can also unlock the possibility to streamline many of them. So that's where the premise came from there. So on the next slide, kind of shows what we're seeing here in our use case. It's kind of two things working in a circular motion. So you're seeing our two tracks moving in parallel, continuously informing each other. So we're approaching our use case that way with the inner track as the foundation, where we built everything from and talked about on the previous slide, and showing that that ecosystem can coordinate these complex decisions across prescribers, pharmacies, intermediaries, and the REMS administrators using those same patterns. So we're currently seeking health systems and EHRs today. So if you know any of those, or that's our hopes, is to bring health systems or an EHR in to help participate in a real world pilot. The outer track is what's being informed by the inner track and opening it up into the pharmacovigilance and risk management side of things. So we're elevating our technical work, stakeholder engagement, and policy considerations and cross-standard alignment to figure this out. I think the important part is that the outer track is not separate from the inner work. So the inner pilot is really going to help continue to get us where we're going. I threw in this slide, Pat, as a surprise to you, but on the next slide, I just wanted to show you this is all we're working on. It's very complex. I'm not going to go into that today, but if we are setting up new call cadence, we will email folks accordingly so that you can join if you're interested. And so we'll follow up with that afterwards. I'm going to send it over to you, Pat, now so you can run us through where we've been and where we're going. Thanks.
Yeah, thanks, Kelly. I'm Pat LaRock. I'm the technical lead on the work on the use case from the MITRE Corporation. So what we're doing and how we've had some of our last tests is building up the use case, building up the to inform the IG, to be able to test the real end-to-end. Well, the ultimate goal of what we're trying to approach here now is a real-world pilot for end-to-end, including all the different stakeholders from the prescriber and the patient all the way to the pharmacy and the patient again when the medication is dispensed to the patient. So that's our goal and what we're trying to do. We took part in the Connectathon from this past January or last month, basically, where we were tried to build upon what we had done before. And we were able to demonstrate the full workflow. We have recordings available. We can add a link to the chat here to where we have all those. But we were able to get good recordings and demonstrations of everything we had done in the past, which included requirements discovery of what the REMS program involved and form completion. So you'd have forms using Smart and Fire to launch into, to have auto completion, to be able to really capture everything that was needed from the prescriber and the patient as part of the REMS program. And then last year we had worked on demonstrating passing the medication on down to the pharmacy, and we were able to demonstrate some of that with our test applications as well, showing some of the NCPDB script messaging that happened there. And so we were able to build upon that a little further by having some error communications. So if you're in the not happy path where a medication is prescribed, gets down to the pharmacist, the pharmacy tries to verify that the medication can be dispensed, but when they try to retrieve the RDA, the REMS dispense authorization, they're not given it. There's an error because some of the forms haven't been completed upstream. The patient or prescriber haven't completed what's necessary. So we added some communications in there that go back to the EHR to notify the EHR that you need to go ahead and complete some of these things that are required. So that was it. We were able to successfully show that. And finally, we had a good collaboration with the PASIO project. PASIO is post-acute care interoperability. So what they deal with is really transitioning of the patient in and out of a hospital system or into different environments like skilled nursing facility or onto the primary care physician afterwards. And so they have what they've built up is this this document that as a bundle that contains all of the medications for the patient, and we were able to demonstrate that we could retrieve that from their EHR systems, process that, and then send off through our REMS, using our CDS hooks for discovery, and find that this prescriber hasn't been registered yet, even though the patient is already on a REMS. So we're able to show that we can complete the workflow and the handoff from a hospital system onto like a skilled nursing facility, for example. So that's where we went from there. Any questions?
Fantastic.
All right. We would encourage folks to put questions into the chat if you like, and I will be sure to pose the questions to the presenters at the appropriate time. And so feel free to use the meeting chat for that. And with that, I will say thank you very much to Kelly and Patrick. Thanks for joining today, and thanks for your work. We're excited to see the PVRM use case continue to evolve and move quite rapidly. So thank you for that report out.
Okay, great. And Pat, would you put your email in too? I didn't.
Absolutely.
All
Very
right.
good.
Thanks.
Okay, and so now I'm very pleased to introduce our feature presentation. Emily Bordelot and Akash Rampersad are two of the leaders in the GenomeX Pathogen Genomics use case team. And they will share, of course, more about the work that they're doing and the team that they've assembled around them to do this interesting work. But this is our exciting presentation for today, and we will pass the mic over to you, Emily.
Great, and hopefully the screen sharing should have already started, so that way you can see.
Can confirm.
Your practice. Awesome. Yeah, well, thank you so much for all of those who were able to join us today, and for all of those who will be watching this back at a later date. Please feel free to reach out if you have any questions. As BVO said, my name is Emily. I received my PhD at McMaster University in biochemistry, and I have a very interesting career in that I've been able to do my research across the entirety of a data lifecycle. Started at the bench, and essentially I was tired of my data being locked in silos. And so that's really what inspired me to join the efforts at CodeX and lead this use case. And I also now have started my own consulting company to create an ecosystem for actionable knowledge exchange, LuxBio. And so I'm really excited to share what the team has been working on for this use case, and then answer any questions anyone might have. And then hopefully some of you today will be able to join us in these efforts, whether now or further down the line. And so to kick things off, pathogen genomics interoperability is increasingly important for global health. Infectious disease remain a major source of morbidity and mortality worldwide, and we're really seeing a clear shift in how pathogen genomics is being used. Historically, sequencing was reactive, performed primarily to confirm suspected outbreaks, and today we're moving towards more of a proactive surveillance, where sequencing is used routinely to detect transmission earlier and to support ongoing monitoring. But what hasn't kept pace with this shift is the way that we're communicating and structuring pathogen genomic data. Most existing health standards, including those that have been developed within HL7, have historically been centered on human genomics and clinical care. And that framing, while works well in clinical settings, it does create friction in pathogen genomics, especially in a One Health context, where the subject of an observation might be an animal, a food product, or maybe an environmental sample rather than a patient. And so with that, for the presentation today, I've actually put together a number of questions to engage with the audience in real time and kind of see who we have in the audience, gather everyone's different experiences, and maybe make it a bit easier to have a conversation. And so BVO, if you don't mind, I messaged you a link for the slide, if you can post that in the chat. But for those of you, depending on where you're joining, you can scan the QR code. And our first question for the COP today is, what perspective best describes how you usually work with pathogen genomics data? As I said for myself in the introduction, I've existed across this entire span, my area of research was in antimicrobial resistance. With my PhD being in biochemistry, I was really interested in learning about how bacteria communicate with each other. And of course, fast forward to now, I'm trying to help create better systems so us humans can better communicate and help fight infectious disease. And so a lot of you are saying that you're with best perspective is within the data standards, health IT. Totally cool. You're not currently working with Pathogen Genomics, here to learn. Super excited to see that. I think a huge part of this work, I'm really excited to be operating in the middle of so many different groups of people, some who aren't familiar with HL7, some who aren't familiar with pathogen genomics. And so it's really great to see some representation for those who are willing to learn. And, we all have something to share in terms of a perspective for data standards and breaking data silos. So pathogen genomics is generated across many different settings, some of the examples I had on the first slide. And that same data may move from laboratory workflows into public health surveillance systems, and eventually into national or global repositories for reuse. And now in practice, that movement is rarely seamless. Even when sequence data itself is high quality, essential contextual information, such as where a sample was collected, the host that it came from, how it relates to other samples, is often inconsistently captured. And when that context is missing, data can become difficult or impossible to reuse downstream. And so this is why interoperability challenges in pathogen genomics don't emerge at the point of analysis alone, but instead at the boundaries between systems and sectors and depending on the use. So one of the most useful ways to understand these challenges is through the lens of the data lifecycle. Pathogen genomics data doesn't live in a single system or a single moment in time. It moves from sampling to isolation, to sequencing, analysis, and often into downstream surveillance, research, and decision-making. Importantly is to remember that this process is linear. A single sampling event may produce multiple samples, and then those samples may yield multiple isolates, which may then be sequenced into multiple libraries. These one-to-many relationships are fundamental to pathogen genomics, but they are often difficult to represent cleanly in systems that were designed around more simple linear workflows. And so as a result, data is frequently forced into these structures that don't reflect how it was generated, and it makes it harder to interpret and reuse later. And so for our next question, I know I've phrased this, where do you often see pathogen genomics data breakdown across the life cycle? But considering we do have a huge portion of the audience that don't necessarily work in the pathogen genomics space, I want you to reflect on the work that you are doing, whether that's in human or other areas of research, clinical settings. Where do you often see data breakdown across the life cycle? Is it where the point of collection or sampling, laboratory processing, when the metadata is missing or inconsistent, or when data is handed off between systems? Or maybe problems only become visible downstream when you're trying to interpret or reuse this data. And so it should still be the same link or as you can scan the QR code to put some input as to what you experience. I know for myself, if I'm thinking back during my PhD, it's definitely, okay, someone already agrees, you read my mind, is the metadata missing or being inconsistent, not having the same means even though to describe a similar or the same concept, whether you're in biochemistry or a chemist. Sometimes we talk about the same thing using completely different terminology. And then it just makes it that much harder to communicate if you don't have some kind of like shared language, or there maybe aren't ontologies that can help bridge these different groups. Some of you have then also said when data is handed off between systems or organizations, yep, it's depending on who's worked with that data and how they've decided to format it. and then handing it off, it's always hard to kind of interpret what that original meaning was. All right. And so this becomes especially clear when pathogen genomics data is considered alongside clinical genomics. And so I've presented here kind of a side-by-side comparison. Clinical genomics workflows are often centered around individual patients and relatively stable care contexts, where pathogen genomics, by contrast, is inherently population-oriented and dynamic. The data may be reinterpreted as new context emerges. The downstream value often depends on aggregation, comparison, reuse across sectors and over time. And many interoperability models that were designed with patient-centered clinical workflows in mind, they don't always make explicit the kinds of contextual relationships that pathogen genomics data depends on. And as I mentioned earlier, my own perspective on this comes from pathogen genomics work in antimicrobial resistance, or AMR. And AMR is an important part of the broader pathogen genomics scope. And it's a useful lens for showing how pathogen genomics data needs to remain interpretable across domains that don't naturally intersect, especially when gaps in structure or context limits the reuse, even if the sequencing data for pathogens are of high quality itself. So drawing on that experience, I'm going to use the GRDI AMR contextual data specification as a concrete example of how upstream data harmonization can support data interoperability across the data lifecycle in pathogen genomics. For some context, GRDI is a Canadian genomics data initiative that supports multiple pathogen genomics efforts, including antimicrobial resistance and COVID-19. And across these efforts, GRDI focuses on defining shared expectations for how contextual and laboratory data are captured and structured across human, animal, food, and environmental sectors. And I'm using the GRDI AMR spec here as a practical illustration for upstream schema that can help reduce downstream curation burden and support reuse across context. And on the slide here, which you can actually click on your screen, are just some resources for the GitHub for the GRDI AMR, as well as the specific One Health data specification, if you'd like to take a look at that later. Then, of course, BVO, I can't remember if you mentioned this, but the slides will be available post the presentation as well. And so now another question for the audience. In your current workflows, where does most of the effort go when it comes to data quality? We're looking at either enforcing structure and validation at the point of data capture, or is it more the cleaning, reformatting, or interpreting data downstream? It can be a bit of both. It's also project dependent. A mix of both. Yep, I hear that. Data quality might also not something you are directly working on. Awesome. And so it does seem like the majority of the group is a mix of both. And so whether it's enforcing structures, with invalidation, or the reformatting, that data quality is going to be critical. And so a key design principle illustrated by the GRDI example is enforcing data quality early. The critical contextual fields must be present before data can move forward. And negative data, cases where pathogen is not detected, it's captured explicitly so that population level analyses are possible. Now, up to this point in the presentation, I've focused on why pathogen genomics interoperability is challenging, particularly how issues around structure, context, and reuse emerge across the data lifecycle. And so now the natural next question is how standards-based approaches can help address these challenges and how different standards operate at different points along that life cycle. And at a high level, the GRDI AMR contextual data spec shares similarities with standards for those in the CodeX community like MCode. This similarity isn't about domain or implementation, but the intent. In both cases, the goal is to distill complex laboratory or clinical information into a consistent, usable form. And systems like this are designed to do two core things: standardizing information coming in from different sources, and they make expectations explicit so issues can be identified before the data is shared or reused. And so this kind of upstream structure is especially important when data is generated by many organizations that may collect and document information differently. And with that, I'm going to pass over to my colleague, Akash, who is going to focus more on the fire aspects of the data lifecycle.
Hello, everyone. I'm Akash. I'm a bioinformatics software dev working at Oak Bioinformatics. My work mainly focuses on clinical and public health genomics, particularly using FHIR and interoperability standards to translate these complex molecular and pathogen sequencing data into standardized shareable formats so that we can implement them properly into clinical decision support workflows and we can affect public health decision making. To properly understand how fire will help us in the data lifecycle, we must first understand the different parts of the cycle that fire interacts with. And to generalize the data lifecycle into two main parts, there are two states of data that exist, either data at rest or data in motion. Fire is mainly about data in motion, but we'll get more into that later. For now, I'll build upon the example that Emily brought earlier with the GRDI AMR specification. This is a specification that belongs more upstream than fire, and it defines what pieces are required and how do they need to fit together before the data is exchanged. This means that it focuses primarily on how data is structured at rest. It defines what information needs to be captured, how that information can be validated or should be validated, and how relationships between data elements are represented consistently. Consistently being the key term here. By enforcing structure and validation early at the near the point of data collection, the JRDI AMR helps ensure that data is well formed long before it needs to be mapped and exchanged into a fire-like format. This upstream consistency reduces downstream burden and helps data implementers and data exchange coders like myself with problems such as missing fields, inconsistent terminologies, ambiguous relationships that don't disappear during exchange, but they just proliferate and get more and more problematic as the data pipeline goes on. To help kind of solidate or concrete this into a real world example, I'm going to bring in the data harmonizer tool, which is part of the GRDI AMR specification or works with it. This harmonizer tool is a template-driven tool designed to harmonize, validate, and transform biological and clinical data. So it's a data schema that allows, once the data is entered in a familiar format, it enforces validation rules and structural consistencies in the background. So it doesn't do it explicitly, but make sure that everything is structured the same way and it has all the mandatory elements. This upstream harmonization, as I said, helps a lot with creating a reliable foundation for mapping data into exchange standards like FHIR. Because I don't know if anybody's worked with FHIR where you go to put a code in the system and there's no code, so you can't do the codable concept. The data harmonizer helps to ensure that these problems don't arise up in the workflow later. One question I have for the group now is, when you think about standards like FHIR, what is the problem you expect them to solve? What is the golden ticket that FHIR is bringing to the situation?
Yeah, whether it's consistent data exchange between systems or maybe, clear definitions of required data elements. I feel like that's something that I've thought a lot about. And again, FHIR is like I've been a part of this use case. I took the role of use case coordinator, I think almost a year ago, this month, which is super exciting.
Yeah, about a year.
Yeah, I know, right? Sorry, this is pretty. And like as I'm learning more and more about fire and relating it right to where I started in the research laboratory, it's definitely the clear definitions of required data elements is the one thing that stands out is just like that common ground for me.
Mhm.
But majority of our audience tuning in is all of the above.
Yeah, yeah, which, fire is pretty versatile, so it does do all of the above. But for some people, fire is a little bit ambiguous and it can be hard to decipher the implementation guide if you've had a chance to look at it. It's quite verbose. There's a lot of pages, a lot of artifacts, a lot of indexes. So to help with this, I've kind of built an example where fire itself is like a box of Lego bricks. Each resource, whether it be a patient resource, observation resource, or a specimen resource, is a standardized building block, like a Lego block. Having the bricks alone doesn't really tell you what to build or how to put them together, which is, the benefit of Lego and the benefit of fire. Fire profiles now, if you're aware of fire profiles, act like the picture on the box. They give you a clear idea and constraints for organizing and combining those blocks together into meaningful structures, and they allow you to share these structures with everybody across in a almost universal language. Now, to further understand something about fire, we should understand where it fits along in the data lifecycle. Fire is optimized for data exchange. It excels at moving information between systems in a predictable and standardized way. What Fire is generally not optimized for is long-term storage. And I see a lot of people get this mixed up. They're like, okay, well, I just store the Fire artifacts when I do the fetch response or the fetch query. This is usually not a good idea because Fire is, I guess nowadays, generally represented in JSON. JSON is, while flexible and expressive, it's very verbose and memory intensive. There are most legacy EHR systems and lab information systems rely on relational databases like SQL or SQLite files, or they even store information in PDFs or images, which I hear is a big problem nowadays, actually. Because of this, many systems do not actually store data natively in Fire. Fire is the exchange or interoperability layer. You only translate to or from Fire when you want to move information from systems. Instead, data is often curated and managed in internal data models and then mapped to Fire, as I said. And it is really uncommon or highly unlikely to encounter a system where Fire is the storage format, but it can happen depending on the local architecture. Understanding the different stages of the data lifecycle illustrate why metadata specs and reporting standards alone are not sufficient enough to guarantee data usability. Because rather than moving raw sequence files or EMR-specific database files, which can differ greatly in content, Fire enables the seamless exchange of interpreted genomic results. As EMRs are becoming less isolated, data sharing and interoperability are playing a big part in connecting a patient's medical records across the entire EHR, and thereby allowing these disparate data silos to communicate with each other is a very important and pain point for patient care nowadays. Now back to you, Emily.
So thanks so much. Yeah, I want to take a moment to say I've really enjoyed putting this presentation together with you because it's the perfect example of, two individuals coming from very different communities, right? Me with the pathogen genomics expertise and you having so much experience in fire and yet being able to find these common grounds, means of communication, and working towards building something that's going to improve patient care and ultimately be a demonstration of what we can really do when we start breaking down these data silos.
Mhm.
And so with that, we've been focusing on the challenges of pathogen genomics interoperability and how upstream harmonization and standards like FHIR play complementary roles across the data lifecycle. And so that brings us to is now the foundational question of before we can build interoperable specifications, do we have a shared understanding of the pathogen genomics domain itself? And so this is the role of the domain analysis model or DAM. It's not an implementation guide, and it's not a technical specification. When I first started learning the jargon of the HL7 space, I was like, okay, this is essentially a fancy needs assessment. We are looking to build a structured description of the core concepts, the actors, workflows, information relationships, and assumptions that need to be represented for pathogen genomics data to be meaningfully shared and reused. And we're doing this work now because pathogen genomics has reached a point where sequencing can be more routine across clinical, public health, and research settings, but the underlying semantics have not been harmonized. Different groups often mean different things, as I mentioned earlier, by the same terms, or assume different relationships between specimens, analyses, and reports. And so the DAM is intended to surface and resolve those differences before we commit to a technical representation in standards like FHIR. And so the DAM will be intentionally broad. What our working group for this use case is working towards is it'll be designed to cover the universe of pathogen genomics use cases that are within scope now and those that are likely to emerge over the next several years. And so this includes, but not limited to, clinical diagnostics, public health surveillance, outbreak investigation, antimicrobial resistance monitoring, with attention to how data flows from specimen collection through sequencing, analysis, and reporting. Now, at the same time, the DAM is explicitly conceptual, not technical. It doesn't define APIs, message formats, FHIR mappings. Those activities are deferred to later phases of the use case, once there's a shared agreement on what needs to be represented and how different pieces of information relate to one another. So this is going to be the foundation for future standards-based implementation. Once we have that agreement on what a specimen is, how it relates, how we can interpret the results, what flows between actors, those concepts can be mapped to FHIR resources and profiles in a way that reflect real-world practice. And so this is why current planning and executions are scoped to the DAM development. Planning for a future pathogen genomics FHIR IG will begin once our DAM is sufficiently mature rather than trying to do it in parallel. Now, in the meantime, which actually this next point touches on one of the questions that we've already had pop up in the chat, this work is being coordinated with relevant HL7 public health working groups and other accelerators to ensure alignment and avoid divergence. And we don't want to be duplicating or preempting existing efforts. Now, to close off our presentation before we move on to questions, because the DAM is defining the domain itself, the input needs to be broad. There are multiple ways to contribute, depending on what you're interested in and what your availability might look like. We have our editorial board, which will be leading the domain analysis model drafting, synthesis, iteration. The working group as a whole will be reviewing drafts, providing domain insight, supporting validation and readiness for the next phase. And I'm also leading a subgroup for the stakeholder needs assessment, which we will be gathering structured input through surveys, kind of like this. I plan to do subsequent guest speakers in the working group if you were to join, to ensure that the DAM reflects real-world workflows and priorities across sectors. And so all of these contributions feed into a single shared model rather than disconnected artifacts. Oh, I love this. Stay informed as the work progresses. That's perfect, because even if you aren't directly involved with pathogen genomics, just staying informed and understanding what is happening adjacent to what you're looking at, I think is so key for us to improve communication and breaking data silos. No one needs to be an expert in everything, but just that awareness is super important. Love seeing the participation in the working group. Please reach out to me or BVO after the presentation, and would love some support, of course, for reviewing documentation, providing some feedback. Tons of ways to participate. And so thank you so much for chiming in there. And so what I has come here today is that pathogen genomics interoperability isn't just a technical challenge, but it is a shared global one. The data we're talking about moves across labs, sectors, borders, and the standards that support it need to also reflect that reality. And so that's why this is happening within HL7 and why the domain analysis model is being built as a collaborative and international effort from the very beginning. And so with that, I want to thank everybody who's been participating in our working group to date. I hope I have everybody here. A few members have joined recently, and so I apologize if they watch this presentation now or in the future and see their logo missing. Everybody who is participating currently is their information on our public Confluence page. So I highly recommend you take a look to see all of the groups. We do have representation, not just across North America, but also several groups in Australia and in Europe. And so we do have that global perspective because, again, pathogen genomics, infectious disease, it's a global health issue. If I were to refer to antimicrobial resistance, bacteria and AMR, they do not care about borders. And so our data standards and how we share information cannot care about that either. And thank you very much. With that, I will hand it over to BVO, and I would love to take some questions with the time we have left.
Very good. Thank you, Akash and Emily. That was an excellent and helpful and clear presentation. And I appreciate on behalf of the entire accelerator, thank you for your work and for your leadership and for the folks that you've brought together. So we will leave the chat. The chat continues to be open. If anyone has questions, this would be a great time to drop them in the chat. In the meantime, I will remind folks that this session is being recorded and that we'll be posting it to our CodeX YouTube channel, and we will be linking that back to Confluence. So those will be available to you. I think we may also do a follow-up with the folks who registered for the event, because there's a few folks who registered who weren't able to join today. And so we'll make sure that that we send an email out so that they can watch the recording.
Awesome. I see a question in the chat I want to already follow up on.
Great, go ahead.
Having to do with the standards across organizations, joint standards development is incredibly important. So we don't want to be duplicating work globally. And I already touched on this a little bit, but I did want to highlight a lot of my partners that I work with through LuxBiome are global. I actually just at the end of last year finished a 2-month research position in Paris, France. And, being super passionate about availability, data accessibility, just knowledge sharing. I'm very intentional at making sure we can reach as many corners of the globe. And being aware that not everywhere has the FHIR implemented, right? And so that's also something to be very aware of, that the data standards that we're building can truly be utilized everywhere, not just in North America. I saw that you flipped your camera on, Bron, so I'd love to hear any comments.
Yeah, no, I think this is an important discussion. First of all, I want to say kudos to the CodeX group. I've been following you all for several years, and you're doing outstanding. Okay. I lead, I'm chair of an ISO group focused on global genomics and omics informatics data standards across 24 countries. It's going to be 30 countries by the end of the year. It's also very important to me as that chair to make sure we're coordinating with other standards organizations. I don't want to duplicate anything that GA4GH is doing. I don't want to duplicate anything that HL/FHIR is doing. So I always have presentations in my meetings from the HL7 Genomics work group and GA4GH. So my ISO genomics group is very aligned with GA4GH. We look at projects that they're working on and have upcoming. We look at ours, we align, and we do joint development.
Yeah.
And this is not a criticism, but it is what the industry is feeling, that FHIR standards and FHIR genomics, it's like every time we get a presentation, and they're great presentations, people don't know what to do with it, who are outside of the HL7 community. And once again, I'm not trying to be critical. I'm trying to provide feedback that will be helpful. So how how does HL7 and CodeX, which is a great use case, bring people into this more simply? So that's one question. Question number two, I've had this come up in Texas. I'm sorry, I live in Texas, but we are funding major cancer research, dementia, and mental health programs as taxpayers into the tens of billions of dollars. Epic is the main system being used across Texas. I reached out to Epic and I'm like, do you have HL7 fire genomics in your system? No, no. So the only way for somebody using Epic system, which is the most widely used system in the world, to get a pharmacogenomic test is to order it separately and then bring it into the Epic system. And I'm still not sure about the interoperability. I have a meeting, me and a couple of people from NB Anderson and UT University Health Science Center are going to have a conversation with Epic. So they're on an old version of HL7 Fire.
Yeah.
And so, once again, this is not criticism. This is a passion in me. I want to
I can
solve
feel it. Yeah.
these problems. Anyway, so there I
So
am.
no, I love it. I will say the second question I think is more than just like with respect to this use case, right? And so I think having that could be its own presentation and conversation. But what I do want to touch on that in terms of that first part,
Yep.
the reason Akash and I collaborated on this talk and how it was structured is because those specifications, the tools that we shared today, are what is primarily used within the research community that have zero clue what HL7 is, right? And so working with my colleagues at Simon Fraser University or McMaster University, right? That's the entry point for them, which is why for the first community of practice call, we took this approach of how can we identify something tangible for those groups who don't know anything about HL7 or FIRE or any of the accelerators. Let's give them something to identify with in the data lifecycle and then show them where they exist and where their data exists compared to when the exchange needs to happen, right? And so those are the types of messages and type of presentations we're really going to be focused on, because the primary goal of the domain analysis model is to bring these groups of people together, right? And a great example, I was in Lyon, France for the One Sustainable Health Forum just this past November, a global event. And it was such incredible feedback I got and excitement about this space from, again, people who had no idea what HL7 was, but were passionate about pathogen genomics, and those who actually did know and were a part or are a part of the HL7 community, right? And so the opportunities are there. I also work with GA4GH, would love to connect more offline so that way we can have a consistent means for us to remain aligned. I'd love to comment more, but I want to make sure we have time for other questions. But thank you so, so much. I love the passion and cannot wait to chat more. Craig.
Thank you. Thank you. Great presentation. I wanted to just check on something. I think what I saw is that you're looking to do the enforcement of data quality with the data at risk and typically before sharing. If I got that right, that brings up a couple of things in my mind, one of which is what the impact is to imperfect data that is at rest, but still may be useful to someone else, and whether those quality checks would inhibit sharing of the data, as opposed to sharing data and then doing the validation on ingestion, where the receiving system can evaluate not just the quality of the data, but the utility to them and accept even imperfect data. And then also how it relates to the fact that given that FIRE, as you say, is not a long-term storage model, and particularly existing systems and legacy data are geared towards local requirements, how you see the application of data quality validation at rest being scalable and shareable tools and all of that stuff where the data model itself may be a little bit different.
Awesome. Yeah, there's a lot there. I'll invite Akash as well to maybe comment on some of the technical part, but to touch on your first part, Craig, trying to make sure I remember a bit of the question.
Yeah,
In terms
imperfect
of
data.
Yeah, imperfect data. Thank you. Yeah, there's never going to be perfect data, right? Which is why having those mandatory fields and trying to capture at least a minimum amount, right? For that context, because it's not something that I have a primary experience with, but what I could imagine is if you were to wait until after to do all of that validation, their context might be lost, right? So I would imagine, Akash, I don't know if you would agree, but having a certain level of validation, you need that consistency prior to that data moving so that the contextual information isn't lost. What do you think, Akash?
Yeah, well, it's a lot to unpack in the question, but what
Right?
my mind goes to immediately is that an ounce of preparation is worth a pound of cure. Where that if we leave it for too long in the data stream or the life cycle, we might run into more either regulatory problems or even infrastructure problems where we're trying to upload something into, like, let's say a JAX, a genomic access server, and we don't have the required fields, like we don't have genome build, right? So now the workflow breaks down, so that could be a problem. But again, it's a kind of a passing of the hot potato of like whose onus is it to have to make sure that all the metadata is there? And I don't think there is like a correct answer. I'm sure I'm not answering the question completely, but I'm trying to like decipher the entire thing. Because
Yeah, I think it's important to just about how we view the data lifecycle, though, right? Because I think, in my experience, anyway, a lot of actors in different parts of it, right? They are just they're existing in the bubble, right? And so I think we also need to work on when we talk about breaking down data silos, it needs to be about sharing that ownership of the data, right? Not just I worked on this stage of it, now I'm passing it forward, right? As I said before, not being an expert in everything, but at least having that awareness as to what's going on in the data lifecycle process. And so I think it's the bigger picture of shifting how we look at how we share data and what needs to be captured. And of course, with that, though, there's, as an academic, funding constraints, right? And so sometimes you're limited by the tools you have access to, which is its own separate problem. And if I continue down that rabbit hole, I could go on forever on terms of the limitations that are just outside of our control. But that might have been a roundabout way of answering your question, Craig, or rambling thoughts, but I hope that.
Yeah, no, that's good. Thank you. And I think the dam is actually exactly the right place to start for a lot of those things. So thank you.
Yeah, and I'm really
thank
excited
you.
to work with the public health group. Really appreciate the time they've given so far to meet with us and share perspectives. And, our editorial group has really rounded out, and we're excited that next week we'll be going to the steering committee to get permission to go from planning to execution phase, which I am confident will stream. Yeah.
Yeah,
Cannot
I will
wait.
say the idea of imperfect is better than none drives strictly from public health experience where, beggars can't always be choosers, and having some data, even if it's not ideal, may in fact be better than having nothing at all.
Yes, imperfect, but usable, right? I guess imperfect. How do we define imperfect, right?
Yeah, not non-compliant, but useful is still useful.
Exactly, 100% agree. We have 3 minutes left, but I do see a hand raised, Braun, so I'd love to hear what you have.
So, all right. So we have all these moving pieces in the omics, genomics landscape, and being able to use that information downstream in electronic health records and clinical trials. Like we all want to do that. That's why we are all here.
Why we're all here.
It is. It's why we're all here. But the problem is the thing that I'm struggling with. There is no funding for cross STO coordination. So when I do cross STO coordination, which is a lot of work, it's on my own time. There is no organization funding. It's like I think all these STOs and CodeX should be locked in a room for 5 days with whiteboards in a workshop and start developing a framework. There needs to be some sort of a common data model that all these things plug into. Um, anyway, I'm just putting that out there. And I am out looking for somebody to fund a workshop like that. So I just want to put that bug in y'all's head.
Yeah, and it's brilliant. It's
And
what's
it's
needed,
lock
right?
the door. Lock the door. Nobody is getting out of here.
Yeah. Well, I would counter that with, and again, from my experience as an academic, right? When I look at the funding calls
Yeah.
for research grants, right? I still see a majority geared towards the discovery and development, which is very
Yeah.
important. But
Yeah.
antibiotics, because I'm AMR researcher, I know there's 60 seconds,
Yeah.
so I'll make this quick.
Yeah.
It's important. It doesn't matter how many new antibiotics or vaccines we develop, if we cannot share the information and
Yeah.
have our patients informed and trusting us with these decisions,
Yeah.
matching with clinical trials, then it doesn't matter how much we develop because it's not going to be able to
Right.
reach the person
Right.
it needs to, right?
Yeah.
And so you're absolutely right, but it's bigger than this in terms of what
Yeah.
needs to change
Yeah.
for how we fund these initiatives and recognize their importance.
Yeah.
So
All right.
with that, BVO, I'll
Thanks for
give
taking
you
my
the.
comments and questions. Okay, I'll
Yeah,
shut up.
that's perfect. No, you're good.
Not at all. All right. Well, thank you very much to our presenters for all of your work in leading this work, but also in assembling your thoughts and bringing a presentation together. Thank you to our engaged audience for asking good questions and for spending your time with us today. If you've heard anything here that's interesting to you and you'd like to pursue more engagement with Codex, please reach out to myself or to any of my Codex colleagues. Also, if you think of anybody that would benefit from seeing or hearing this presentation, please share this along to them. And with that, I wish you an excellent day, and thanks again to everybody.
Thank you,
Very great
everybody.
meeting. Great meeting.
Such a pleasure meeting you all.
Yep.
Thank you.
Yep.
Happy Friday.